Okay, my presentation is completely different. I'm from a biomedical science background, and then I'm a biomedical scientist. So, as I can see, like most of the presentation here is from the science and technology, but not from the biomedical science. So, I'm a biomedical scientist. My name is Susbin Wagle, and I'm working in Cutting University in a biotechnology and drug development research laboratory. We stay in Perth, WA, and today presentation is all about the bile acid based micro nano capsule for the targeted oral delivery of Provical. So it's completely about the drug delivery, drug formulations, and then treating the and metabolic disease and all those things. So it's completely different as I can see. Um, Talking about our research, it's all about the drug formulation, preparation, and delivery. And here we are using the drug Provical that has the anti-inflammatory and antioxidant capacity and has the potential to prevent the diabetes mellitus. As we all know that diabetes mellitus is a metabolic disorder characterized by the high blood sugar level. But the current problem with the diabetes mellitus is that till now there is no effective medicine to prevent the pancreatic beta cells from the inflammation and the oxidation. So who's inside the PB has this capacity, that's the Provical has this capacity to prevent the diabetes mellitus from the anti-inflammatory and antioxidation, and we're using the micro nanotechnology for that objectives. And therefore today my talk will be about the drug formulations, design, and then applications in the diabetes mellitus by using the microencapsulation technology. Like now I like to start my presentation. As I told, like diabetes mellitus is a metabolic disorder, and it's not confined in one particular place or one particular specific in one particular country. The incidence is all around the world. Talking about the diabetes mellitus more, it is a metabolic disorder characterized by the high blood sugar level, and then it has the various complications from such as like later on at the early stages that it can be people can suffer from the different kinds of complications such as neurodegenerative disorder, and then the heart disorder, and then the kidney dysfunction and all those disorder. And talking about the incidence, it's around 470 million people are suffering from the diabetes mellitus, and this figure predicted by 2045, the populations people suffering from the diabetes mellitus will be around 700 million. And then each year 4 million people are dying from the diabetes mellitus, and the cost to prevent the diabetes mellitus is around $730 billion every year. And more than 21 million people are affected by the diabetes mellitus during the pregnancy, and then around 350 million people are at the risk of developing the diabetes mellitus. So this is very hot topic, like as perspectives from the people points of view as well. And as we're doing the research in Australia, talking about the diabetes mellitus in Australia, it is the sixth highest causes of death by disease in Australia, and then it is believe that out of 4 Australian, 1 Australian adults have either diabetes or the pre-diabetic symptoms, and around 200 people diagnosis every week. So the cost to prevent the diabetes mellitus only in Australia is around $14 billion every year. So and around 12 to 14% pregnant women are suffering from the gestational diabetes. Gestational diabetes is kinds of the diabetes where the pregnant women suffer from the diabetes. So that kinds of diabetes we call as the gestation diabetes. And broadly, the diabetes can be divided into type 1 diabetes mellitus and type 2 diabetes mellitus. Type 1 diabetes mellitus is quite related to the genetic disorder and all those things, whereas the type 2 diabetes mellitus is more related to the lifestyle and then diet and all those things. So later on people can suffer from the type 2 diabetes if they do not modify, and if they have the disbalanced diet and all those things. And the current therapy for the type 1 diabetes mellitus is insulin therapy. Almost all the patients suffering from the type 1 diabetes mellitus depend upon the insulin, and this figure is more than 30% in type 2 diabetes mellitus. And the gestation diabetes patients also the one and only therapy for the gestation diabetes, also the insulin therapy. So what really causes the dysfunctions of the pancreas? Pancreas is the hormone that regulates the blood sugar level. The pancreas contains different kinds of the cells, and one of the cells is beta cells, and the beta cells releases the hormone called insulin, and this hormone is responsible for the regulations of the blood sugar level in our body. So, for example, one of the causes of the diabetes, that is type 2 diabetes. I'm focusing more to the type 2 diabetes rather than the type 1 diabetes. One of the causes of the type 2 diabetes is obesity, and in obesity, the primary causes of the obesity is inflammations. When there is inflammations, this inflammation directly damages to the pancreatic beta cells, that the hormone, that the organ that regulate that release the insulin and that regulate the blood sugar level, and so because of this high pro-inflammatory and the cytokine release, it causes the dysfunctions of the beta cells, that's one of the cells present in the pancreas, and which causes the decrease in insulin secretions, that the insulin is the hormone that regulates the blood sugar level, and increase the insulin resistance and causes the lipid accumulations, and finally people suffer from the type 2 diabetes mellitus. So there are many anti-diabetic medicines in the market, but the current anti-diabetic medicines and medications are mainly effective in managing the blood sugar level. So, as I told earlier, the diabetes mellitus is characterized by high blood sugar level. So, the current anti-diabetic medicines are mainly effective in managing the blood sugar level. Either if the people suffer from that, if people have a high blood sugar level, we generally prescribe the anti-diabetic medicines. But most of the people are dying from diabetes mellitus. It's not because of the high blood sugar level, but because of the complications, like the created by the high blood sugar level. That is the we normally call it as the diabetes associated complications. And these current anti-diabetic medicines are not effective in managing the diabetes associated complications, like I mentioned earlier, some complications like the heart disease, retinopathy, neuropathy, and then the kidney dysfunctions and all those things. So these anti-diabetic medications are not effective in managing these diabetes associated complications, and also they are not effective in the hyperglycemia and then free radical. Always there is a risk of the development of the free radical, and then the toxin development, always risks with the current anti-diabetic medicine. And it is expected and predicted that the total number of diabetes mellitus will suppress all the present prediction within the coming 20 years if no novel and effective medications are invented. That means right now there is no any effective medicine to prevent the pancreatic beta cells. Just what we have to understand here is one of the causes of the diabetes mellitus is because of the inflammations of the pancreas, and there is no any effective medicines that has capacity to prevent the pancreatic beta cells from the inflammations and the free radical developments. So the current challenge is that, and we think that Probical has that capacity because it is anti-inflammatory and has potential to prevent the diabetes mellitus. That's why we picked the drug Probical. Talking about the Probical, this drug is initially marketed as an anti-hyperlipidemic drug. This drug is not for the anti-diabetic. This is not an anti-diabetic drug. What we need to understand here is this drug is initially introduced for the anti-hyperlipidemic drug, and this drug is orally administered. But the problem with this drug is, like, it has a low viability. Low viability means low clinical efficiency. Low viability means the drug cannot be absorbed from the body, and then it is a highly lipophilic drug. The other problem is highly lipophilic means that they are not soluble in our gastrointestinal tract. If the drugs are not soluble in our gastrointestinal tract, then the drug cannot be absorbed properly, and then it has a high inter-individual variations. That means that the doses is depend upon person to person. So if some person need a high amount of the drug and some person need the low amount of the drug, so it's have the high inter-individual variations, and it is accumulated extremely in the adipose tissue. Rather than the absorption from the site of absorption, the drug is accumulated in the adipose tissue because of all these things, like low viability, low water solubility, high inter-individual variation, high toxic development, and all those things. After, I think, after the 20 years used in the market, the drug is withdrawn from the market. So the FDA from many countries like the US, Australia, and then many other countries, European countries, Western countries, they withdraw this drug. But the good thing about this drug is that, as I told, it has an anti-inflammatory and antioxidant property. That means this anti-inflammatory and antioxidant property can protect the pancreatic beta cells from the inflammations. But as I told, this has many disadvantages. So this disadvantage can be overcome. Without this disadvantage can be overcome by making new formulations, and this new formulation can be done by using the nano encapsulation technology. And we use this technology to overcome this problem, and then and we think that we can prescribe this the drug to the diabetes patient. So I don't want to describe here more about the value, but just in the brief, what causes the decrease in oral absorption and the viability of the drug? So when the drug is orally administered, any kind of any kind of the drug, when we orally administered, is uh, uh, can you or not, because the internet connection is showing quite poor. Okay. When the drug is orally administered, so there are different enzymes and the proteins present in our gastrointestinal tract that this kinds of in the orally administered drug, like a lipophilic drug that has the low water solubility, like this drug normally interact with this enzyme normally interact with this kinds of the drug, and then causes the degradations of the drug. That means before it's reaching to the target site of the absorption, the drug normally absorbs from the lower intestine. So before reaching to the lower intestine, normally drug interact with the different enzymes, protein present in our gastrointestinal, and that makes the degradations of the drug, as well as the pH environment, the pH present in our gastrointestinal tract, that causes the degradations of the drug. All these substances, like we need to protect the drug from enzymes, proteins, and the pH environment, all these substances before reaching to this target site. This is the small intestine, and the pH of the small intestine is around 5.5 to 7.5. So we need to prevent the drug from in stomach, like in stomach, and from the unwanted degradations. So if we are not able to protect the drug, and this causes the decrease in oral absorptions, because the drug will not come to the absorption site here, and then causes the decrease in the oral viability, or decrease the clinical efficiency of the drug. This is the basic reasons why we need the high doses of the drug, or why the drug are not effective even we are taking every day the same drug. This will be the reason. And this can be overcome by using the encapsulation technique. The encapsulation technique means simply, this is the process in which the particle or dropless substances, that is drug, is protected. We need to protect. This is the active ingredient means drug. We are protecting this drug by using the encapsulation matrix. So the drug is normally protected by a different kind of a polymer, and then when we orally administer it, so it cannot be degraded in the stomach, or the drug is protected from the different proteins and enzymes, and causes and safely reaches to the intestine, the lower intestine, where the site of the absorptions happen. So this is the basic principle of the encapsulation technology. And broadly, encapsulation technology can be divided on the basis of the size nano encapsulation and the micro encapsulation. Nano means smaller in size, that is less than 1 micrometer, and the greater than 1 micrometer we normally call as the micro encapsulation, and we prepare the micro encapsulation. And this is the broadly nano and micro encapsulation. The technology is the kind of the nanotechnology. It's not related. It's a nanotechnology. It's quite related to the biomedical. So what we need to understand here is it's not related to the other like a physics or some atoms or something. It's related to the drug. As I told, why we need the encapsulation of the drug for the protection of the sterilizations of the particles, that is from the protections from the different pH environment, from the enzymes and protein present in the gastrointestinal tract, And then for the targeting of the encapsulation compound to the specific type, that is to the lower intestinal where the absorptions happen. And for the control or the timely release of the core materials, for the control release of the drugs. So the drug is always good for the diabetes drugs. Drug are always good if the drug is controlled and a timely release, because this control and timely release reduce the toxicity and increase the effectiveness of the drugs. So because of this, we are doing the encapsulations of the drug for these reasons. And for encapsulation, we use the sodium alginate. This is a polymer that's protect the drug from the outer layer. That's the coated materials. And why we choose this sodium alginate is because it has a biodegradable, biocompatible, and a hydrophilic and the protective characteristic, and has shown the excellent compatibility, stability, and the delivery potential in the targeted drug formulation, and then and has support the control and the timely release of the drug as well. And it is all maintained the pH and the release kinetics. That the pH release kinetics means normally this kinds of the polymer protect the drug from lower pH, and then when the drug reaches to the higher pH, normally the outer layers break down, and the drug absorptions happen. So this is the absolute and good for the drug like the Probical, for the protections from the unwanted degradation. And one important thing about the lipophilic drug like a Probical is they have the low permutations. That means they cannot penetrate from the site of the absorptions. So to facilitate that penetrations and increase the absorption, we use the bile acids. These bile acids are naturally produced inside the human body, and has the drug permutations across the intestinal epithelium cells, and also this bile has the membranes stabilizing molecules. So we also call as the membranes stabilizing molecules, and it facilitates to uptake the drugs and then and support the absorption of the drugs. So because of these characteristics, we choose the bile acid to mix with the drug for the improvement of the drug absorption, because this has the good potential for the drug permutations across the intestinal epithelium. And there are different kinds of the bile acid. The one we choose is the lithocholic acid because it has a versatile structure and then a high structural rigidity, biocompatibility, and biodegradability as compared to the other bile acids. Other bile acid has also this property, but it's lithocholic acid LCA, that's we call as the LCA in short form. It has more this kinds of property, and it has a high hydrophilicity as well, that makes drug more soluble. And then the conjugations of the lipophilic, and study has the conjugations of the lipophilic anti-cancer drug like tamoxifene conjugated with the LCA using amide pH stability and better absorption. These are the some of the useful of the LCA done by the other groups, and it has shown the anti-inflammatory property and formulation stability in atherosclerosis also. Because of this, all these characteristics, which is okay, bile acid can be the one that can facilitate the or can improve the permutations of the drug. So which is the bile acid, and the method we use is the ionic gelation vibration jet for technology. This method is the combination of the physical and the chemical microencapsulations, because it's operated or use both mechanical as well as the chemical processes. And normally in this method, the product this is the product means like a drug, and then the drug to be encapsulated is mixed with the polymer. And that's the polymer is the sodium alginate, and the put into the pressure bottle. This is called the pressure bottle. And the polymer, this mixture is forced into the bead producing unit by the air pressure. So from here it goes to the here with the help of the air product, with the help of the air pressure, and we call it as the bead producing unit. The liquid then passes through the precisely drilled nozzles and separated into the equal size droplet when existing the nozzle. So from here, normally from here, from the bead producing unit, this is the nozzles, and the droplet passes from the nozzles and produce this is the capsules. Actually, this is the microcapsule. So they produce the equally sized drain and the equally sized droplets, and which we call as the capsules. And these droplets passes through an electric field. So this is the electric field. between the nozzles and resulting in the surface charge. This electrostatic repulsion force disperse the bit as they drop to polymerize electrically grounded. So when the nozzles charge, so they cannot collide with each other. That means you can see the disperse. So these particles are dispersed from each other. So that's why we call as the jet flow as well. You can see like a jet, like this the method is called because of that. This flow structure, this the droplet flow is called as a jet flow technique. And it can produce and this so it can produce produce the spherical shape microcapsules, and we collected this microcapsule in the polymerization bath, and this also electrically grounded. If it is not electrically grounded, then it forms rather than forming the microcapsule. So this bath should be electrically grounded, and we normally collect it in calcium chloride, and the calcium chloride is a gelatin bath actually. So this is all. This technique is also called the artificial cell microencapsulation technique. This technique is initially introduced by the Professor Thomas Chang at the Wagle University, and we have the collaborator. He's one of our collaborators as well, and Dr. Hani Al Salami. He's my supervisor, and he introduced this technique, this kinds of the techniques in Australia and in our lab. Actually, we are only one who produce the microcapsule by using these kinds of technique in Australia. So talking about the results, I don't want to go, like detailed methodology because got a time limit and all those things as well. Talking about the results, firstly, what we did is that we prepared the microcapsule by using the prepared microcapsule is examined by using the microscopic examination. So to know the shapes of the microcapsule we normally produce, is the capsules spherical shapes, or like what shapes. So we did the microscopic examination, and this is the microscopic examinations of Pb assay. So we produce two kinds of the microcapsules, and just to understand, we produce one is probical and sodium alginate. The next one is we produce the probical, sodium alginate, and lithocholic acid capsules. So we want to know what our basic objective is. We want to know the impact of lithocholic acid on probical microcapsules, because there are a lot of studies already done using the probical and the sodium alginate, and we want to add the LCA, because the drug, this probical drug is highly lipophilic, as I told, and then we want to know the effect of the LCA on the Pb microcapsules. So we produce two kinds of the microcapsules, and is that the LCA on Pb microcapsules doesn't changes any there is no any changes in the morphological structures of the microcapsule. That means the spherical shapes and the uniformity and all those things. So there is no impact of the LCA on the Pb microcapsule. First thing what we try to find from here is that to know the impact of the LCA microcapsule. Furthermore, and we did the SEM, that is the scanning electron microscopic examinations. In SEM examinations also, what we found is that both the microcapsules, that the Pb and assay Pb and assay and Pb LCA and assay microcapsules. So this is Pb assay, and this is Pb additions of the lithocholic acid, assay and LCA microcapsules. So both the microcapsules behave the spherical shapes, and then like when we do the surface examination and analysis, what we found is that there is not there is the small depositions of the granules. You can see here they are the depositions of the granules on the surface of the microcapsules, on both kind of the microcapsules. So to know what actually that the granules is, so we just want to know the surface. The microcapsule is a little bit crack in both kind of, here you can see on the surface and the microcapsule is crack and the deposition of the granules. So to know what actually that granules is, we did the EDXR analysis. That is the for the surface character analysis. So what we found is that the granules present on the surface of the microcapsule contain the high amount of the sulfur. So these granules contain the high amount of the sulfurs, and to understand this, the probical there is so we prepared the microcapsule by mixing the sodium alginate, lithocholic acids in the polymerization, and only the drop, that is only the probical contain the sodium sulfur. So only the that means that granules present on the top of the microcapsule is actually the drug. That's the provicle. So what we found is that on the surface of the microcapsule, drugs are present. Inside the microcapsule as well, the rocks are present on the surface of the microcapsule as well, which we found by using the EDS analysis. And further to check the compatibility of the microcapsules, we did the to check the physiochemical compatibility of the microcapsule. We did the DSC and FTIR analysis. DSC means differential scanning calorimetry, and FTIR means a Fourier transfer infrared spectral analysis. What actually the DSC measure is, it measures the thermal profile of the molecules, changes along with the temperature over the time. So we are the first one to measure the thermal capability of the LCA, and LCA would examine together with the Pb microcapsules. The temperature here, you can see in this table, the temperature here is the melting temperature of each corresponding compounds. So what we did is, we found the melting temperature of each compound first, and the melting temperature of the Pb is 128 degrees Celsius, and then all other compounds LCA would find the melting temperature, and then we mix all these three compounds at 2 melting temperature. We have the 3 compound, but we found only 2 melting temperature. And this 2 melting temperature, one is the provicle. You can see, the melting temperature of provicle is 128, and here the melting temperature is 129 and 201. So what we found here is, LCA and SA, they have the similar melting temperature, and they actually interact with each other. This interaction make capsule more stable. What we understand is, the LCA make capsule more stable, and it causes the interaction with the polymer, but this interaction doesn't make the impact on the thermal capability of the drug. That means you can see the clear melting temperature of the drug. So this interaction doesn't make any interaction, so it doesn't make any impact on the drug thermal capability. That means physical compatibility of the drug pre and post microencapsulation. That similar pattern was observed after the post microencapsulation. We prepare the microcapsule, we dry it, and we green it, and then we measure the thermal compatibility of the microcapsule as well, and then this similar pattern is observed. So there is the interaction between this bile acid and the polymer, but this interaction doesn't change any impact on the drug thermal capability. And the similar pattern observed in the FTIR analysis. This FTIR analysis is more about comparing the chemical compatibility of the compound. So DSC analysis check the thermocompatibility of the compound, and then FTIR analysis check the chemical compatibility of the compound. Overall, this FTIR and DSC compatibility checks either the microcapsule we prepared are compatible or not. So what we found, this is similar to the DSC. This is the corresponding spectra of each compound. So the Pb powder, they have the 3 different spectra, and then this is the mixture of all 3 compounds. And we found that LCA and SA, they interact with each other, but this interaction doesn't make any chemical any impact on the chemical compatibility of the drug, similar to the DSC results. That means pre and post physical and chemical compatibility of the drug is maintained throughout the process. What we can conclude from this result is that. And further, we investigate the effect of the bile acid on the encapsulated drug. That's either the drug content will change or not. Either the microencapsulation efficiency will change or not after the additions of the LCA, that is the lithocholic acid, on the Pb SA microcapsule. So what we found is that production yield, encapsulation efficiency, drug content doesn't impact by the additions or incorporations of the LCA on Pb microcapsule. That's what we found. And then further, we want to investigate the effect of the LCA on the on Pb microcapsule. What we did is we tried to know the soiling behaviors of the microcapsule. So we choose 4 different pHs. This pH reflect the pH of the gastrointestinal tract. So pH 1.5, pH 3, pH 6, pH 1.5 to 3 is the pH that reflect the pH of the stomach, and then from 6 to 7.8 that's reflect the pH of the gastrointestinal, the pH of the lower intestine. So we want to see either the microcapsule soil in the lower, if the microcapsule soil and degraded in the lower temperature, then the microcapsules we prepare are not stable. But what we found, the microcapsule is hardly swell at the lower temperature, lower pH values. You can see, like there is less swells, but when the pH increase, the microcapsule swell more. And then this F1 is the PBSA microcapsule, and then F2 is for the this is just a code, and F2 is the PBSA and LCA microcapsule. So what we found is that the additions of the LCA, what we need to note here is the additions of the LCA makes microcapsule less swell. That means it decreases the solubility, and LCA addition decreases the solubility of the swelling properties of the microcapsule. That means it makes microcapsule more stable. If more swelling, then the microcapsule bursts easily. But if the microcapsule is not swell, then it's support in control and timely release of the drug. So we did in two different temperature, and the temperature at 25 degrees centigrade and the temperature at 37 degrees centigrade. And of course the impact of the temperature is also seen. That means when the temperature increase, there is more swelling of the drugs, and then the swelling increase as the pH increase. So to compensate for this swelling behavior, we did the different kinds of analysis. We did the mechanophysical resistance as well. So this F2 and F1, what we found is that PB LCA microcapsules, the additions of the LCA on the PB microcapsule make microcapsule more intact. Means they support the microcapsule from unwanted degradations. That's the impact of the LCA microcapsules. Also, this PB LCA microcapsule support the microcapsule floating. Means if any microcapsule, if the drug has that good floating properties, then it supports the control and the timely release of the drug. And then we measured the pattern of the drug release. What we found is that at the pattern of the drug release in 4 different pH value, there is no release of the drug at the lower pH. That's what we want, because drug is normally absorbed from the lower intestine. That's at the pH value 7.5, more than 7.5. So there is no release of the drug at low pH value. But when the pH value increased, you can see there is almost 80 to 90% release of the drug. But also what we need to notice is that there is less and timely and controlled release of the drug by additions of the LCA. So here we can see, like this is the without the LCA, there is more release of the drug, and the additions of the LCA decrease the release of the drug and support the control and timely release. And this is the basic and important feature in the anti-diabetic medicines. That's what actually we are looking for. And further, we did some biological analysis as well. We want to check the impact of the LCA microcapsules, PB LCA microcapsules on the cells. And so this biological analysis was done on the pancreatic beta cells, that is called NIT. So that means that the cells is similar to the pancreas present beta cells present on our human body. So what we found, what we're trying to find here is, like we did this biological impact of LCA on NIT cells on 2 different environment. One is 5.5 millimole, that is the hypoglycemia at the normal conditions. So at the normal glucose level, and we try to find the impact of LCA at the hyperglycemia. This is the 25 millimole. The hyperglycemia means more diabetics, more stress to the cells. So at the normal glycemia, there is no changes on the cell viability. Means the number of the healthy cells. Viability means number of the good cells after the treatment. So there is no change. This is a control. Here you can see this is the control. This is the health control, means without any microcapsule. So we treated with the PBSA microcapsule, and we treated with the PBLCA microcapsule. So there is no impact on the at the normal condition. But at the hyperglycemia, you can see, like this is the control microcapsules, and this is treated with the PB microcapsule. There is increase at the control microcapsule. There is a low cell viability. You can see here low cell viability, but the viability increased when we treated with the PB microcapsules. Significantly, there is improvement, significant cell viability. And similarly, as in diabetes mellitus, there is release of inflammations and cytokines and inflammations, and then we try to know the impact of this microcapsule on the cytokines as well. So we choose different anti-inflammatory and good cytokines release, impact on cytokine release. What we can find is, like there is this PBLCA microcapsule decrease the release of the cytokines. That is, this cytokine causes the inflammations, causes the decrease in the release of the cytokines. But we need the anti-inflammatory cytokines also that protect the pancreatic beta cells. So it causes the increase of the anti-inflammatory cytokines. Here you can see the additions of the LCA causes the increase of the anti-inflammatory cytokines. Anti-inflammatory means good for our body, but it causes the decrease in inflammatory cytokines, causes the decrease of the inflammation that is bad for our body. So overall, it causes the LCA addition causes the increase in the cell viability, causes the increase in the healthy cells, and it causes the decrease in inflammatory cytokines, and it causes the increase in anti-inflammatory cytokines. And also we want to know the impact of the PBLCA microcapsule on the bioenergetic parameters. That is, the bioenergetic parameters normally helps to know the conditions of the mitochondria on the cells. So what we can we found here is similar to this in the cell viability. What we found here is that bioenergetic parameters, these are the oxygen consumption rate, proton production rate, and extracellular acidification rate, and maximal respiration, and the basal respiration. All these are the biomarkers for the mitochondrial functions. So if the cells have the good mitochondria, then that means the cells can produce the more insulin. If the cell can produce the more insulin, then it can protect the pancreatic beta. I mean, it can protect the it can control the blood sugar level. So good mitochondrial functions is indicated by the additions of the PBLCA microcapsules. Here you can see they are all the biomarkers for the bioenergetic parameters. So all the bioenergetic parameters are improved by additions of the PBLCA microcapsules. So overall, what we can say from this biological data is, like, PBLCA microcapsule protect the cells from inflammations, from the inflammations, and also it improves the bioenergetic parameters. Means it has the good biological property to it has a capacity to protect the pancreatic beta cells from the inflammations as well as from the oxidations. And at conclusion, this our microencapsulation method is effective in producing the spherical shape, size, and the uniform microcapsule that can be seen from the microscopic examination as well. And then the study examining the effect of the LCA on Pb microcapsule on morphology and thermal and chemical stability. That is from the DSC and FTIR analysis. And the additions of the LCA make microcapsule more stable, reduce soiling, and allows the control and pH targeted release of the drug at different pH values. That is from the soiling effect, and then the mechanical resistance effect, as well as the buoyancy test, that the floating behavior of the microcapsule, and the control release all shows that the LCA addition make microcapsule more stable, reduce soiling properties, and support in control and the time release of the drug. And this properties is very important for the anti-diabetic preparation for the better clinical efficiency of the anti-diabetic medicines. And then at last, LCA incorporation enhance the bioenergetic parameter, decrease the inflammatory cytokine, increase anti-inflammatory cytokine, enhance the cell viability. That means it has the good property to protect the pancreatic beta cells, because all we did in vitro analysis in the pancreatic beta cells, and it protect the pancreatic beta cells from the inflammations and from the oxidations as well. So hence, this PBLCA microcapsule is a novel form of the target delivery that shows the optimal excipient compatibility, structural morphology, and homogeneity. And overall, this suggests the potential applications of a PB in the oral administration in type 2 diabetes mellitus, and will in future. And actually, I already did that study, but because of this time limit, I didn't show the animal study. We prepared this. We prepare microcapsules to the anti-diabetic mice, and and we check the effect of this prepare microcapsule, and we got a fantastic result for that. But I didn't present here. And sorry for that because of the time limit as well. And at last, thank you for you are the wonderful audience, and thank you. And if you have any questions, just let me know. Thank you.